about it because a lot of cancer drugs are antibodies. And you also have fusion proteins, which are often, conjugates of the antibodies to the toxins, or it could be a simple conjugation of the protein to some moieties such as pegylation that results in the better off-life of the protein, etc. By the way, pegylated interferon is used to treat, for example, hepatitis C. So there are other modalities as well. These are FDA-approved drugs. But you look at where we are currently, what are the kind of modalities we're going after, really? And still the small molecule dominates. And you would see the difference here, although there are so many small molecule programs out there, but the increased success is coming from using antibodies and proteins. But as you can see, that the reason they work on this is, even though the success rate is getting better with biologics, is the fact that in order to make the drug accessible to the remote areas and rural areas, they can't go to the doctor every day. I think the still preferred route of administration is, if you can get a drug, is the small molecule. However, for technical reasons, we can get small molecule for every target. Right? So that is the reason why I think the biological is becoming more and more important. And also from pharmaceutical company perspective, biologicals make sense for two reasons. One is you get premium pricing than small molecule. Number two is they're not easy to copy, because unlike small molecules, every biosimilar you make or the copycat you make have to be qualified with the proper clinical trials, make sure it works with same potency and same safety as the parent drug. That is why you don't see a lot of generic companies working on biologics, including vector-delivered molecules and fusion proteins more and more showing up. And then you have antisense as well. So there are more and more modalities the pharmaceutical companies exploring. Now, here is the slide, which I think it's like 2014, I think. It's kind of top-selling 30 drugs here. As you can see, that there's still more than half of the drugs are small molecules. But increasingly, there are large molecules, particularly the antibodies and the proteins are showing up, and this continues to be the case. Now, this is an interesting slide. I just found it, I think it's published only last month, where the route of administration seemed to be shifted a little bit. As I said, the oral drugs dominated. But now, people don't mind taking injectables. There are the benefits I told you, but for the patient perspective, it may be a little bit inconvenience to go into the doctor and have an injectable. But on the positive side, for the patient, biologics tend to be safer than small molecule, right? So they're more, interest in taking injectable than ever before. Now, one surprise I have here is, which I surprised when I saw this slide, is this one here. About a large proportion of the increasingly large proportion of the inhalable drugs. You always use inhaler to deliver. Most of the inhalers have either artecortisone or albuterol in it, so just to breathe better. I think, but I see that even the antifungal and even some of the anti-respiratory, like asthma drugs, being delivered by respiratory route, which is a huge change from 10 years ago. So you would expect to see more drugs delivered through the inhalation. It's not an accident. I think the local delivery of drugs to the lung, where the primary infection is the lung, I think it makes a lot of sense. You can even avoid systemic side effects as well. Not only you deliver the drug, very effective in the lung, perhaps get away with a lower dose than give an injection and oral. I think this is a very effective way to treat. There are some cases it's not trivial because, for example, RSV or flu, you have a lot of sputum and mucosal secretion. It may be hard for the drug to get into lower respiratory tract, but the recent studies suggest yes, you can get enough drug even there. So expect to see more drugs delivered by respiratory route. Amazing there are how many different, you also see this delivered through ears, delivered through, directly into the eye. Now, so the prophylactic vaccines, we talked about, vaccines have been the landmark medicines for Merck and, a few other companies, and these are in most cases they're preventative, and you all, as I said, they've taken a bunch of vaccines, maybe perhaps 1/3 or one-fourth of the drugs or the vaccines you all have been given. And so they're tremendously useful to prevent the disease. And the area that's really getting some attention is what we call therapeutic vaccines. In other words, you give this vaccine to stimulate the immune system after you actually contracted the disease, right? So an example I gave is here, for the prostate cancer. It's one example I know of that's been very successful, but there are many in pipeline being developed to treat a disease which you already have. Okay, that's a future for the therapeutic vaccines. I bet there will be a lot more coming forward. Now, there are very few drugs actually targeting the approved drugs, at least the RNA, DNA. Here is the only handful I can find, There are not that many, and these are often the last resort treatment. In other words, there are no other option for these patients, and they're often very involved. Nobody likes to take a viral vector delivered treatment because they're always concerned of this viral sequence getting integrated into the human genome and stays forever. And for other reasons, these are not the most popular way to treat, but there are some out there and people still working. Now, and then you have cell therapeutics. Mostly you all heard of it, stem cell transplantation, which is very common. And there are very few specialized labs receive these applications or the approvals for this. You notice that these are all academic labs or hospitals associated with universities, because these are the ones they isolate right on the site and then inject into the patient, either heterologous or for some other or homologous transfers here. And so it happens very successful in stem cell transplantation for certain indications. It's continued to be. But the new type of stem cell research coming along, which is iPS-based induced proliferative cell-based therapeutics. That's a future, because when you do that, actually the phenotype of the genotype and phenotype of the cells is not the same. So the body reacts as foreign and it rejects it. But the people are working very hard to make sure that it's not rejected. It's a long way out there, but eventually you will have this cell transplantation using the stem cells, and perhaps also organs down the road, but we're not there yet. Now, this is a huge investment in recent years targeting the mRNA. You all heard of antisense, which has some success. There is one drug out there I showed vitrovine, which is directly injected into the eye. It's an antisense. Besides that, there's not that many drugs approved. But there's a lot of interest with new technologies such as siRNA, targeting microRNA came into the picture. There's a lot of efforts in this area. A lot of companies showed up. Again, slow here. The reason being slow is that the issue is not really with the molecule per se. They had ways to really stabilize these molecules, metabolically stable, et cetera, et cetera. The issue is delivery. They always need a lipid to go with it, otherwise they don't get into the cell. And so that lipid combination really often results in the toxicity, safety issue. So there are only two areas this strategy seems to work so far for the reason I just mentioned. One is directly injecting into the site of infection, either eye, vitrovine is an example, or directly into the tumor or any other indication. So topical administration into the tumor. The other area that seems to be attracting a lot of attention and a lot of molecules out there is the delivery into the liver. Because you all know probably this, when you give an injection of RNA, all of it goes into the liver as a first pass, and often it stays there. So if you're treating a liver disease, for example, hepatitis B or NASH or any fibrotic tissue in that makes a lot of sense, But again, even there it's suffering for the safety reasons, because even there you need to use a lipid formulation. There is a technology called LNA technology where you don't have to use the lipid formulation. Those are the molecules that are coming out now, and hopefully there will be a success there. And so it is an area of intense research, but it's moving very slow, but I'm confident that there will be some medicines coming using any of these modalities here. Again, you notice that I'm not going to details of these modalities and the technologies you would find in the internet. Now here is the this slide and next slide actually list the molecules based on treating the RNA either in clinical studies, in various clinical studies. There are, as I said, the one common theme among all these is that either they target to the tissue directly as a direct injection, or they're delivered to the liver. There are other areas where it's still struggling to deliver because of the safety reason. And this is the second slide. You could read it, and if anyone interested in specific examples, I'm happy to discuss this. I have the chance to actually follow through all this as a part of my job to see any of them looking promising so that we can license them in. Now, then you have this new buzzword or new area, microbiome, which is an exciting area because, kind of ignored because what difference gut microbiome might make to the normal health as well as for the disease. And the difficulties here is to understand which of these bugs in the gut are really essential versus those not essential. Also, other difficulty is that why there is a difference in the composition of the microbiome from individual to individual. Is that means anything? So there are a long way to go in my view, but I think though it's the risk or the chances of being successful here is much better than in the siRNA in my view. Now here is it is a publication that came out in BioCentury very recently, I think 2-3 months ago. It talks about that the microbiome treatment can be classified into 3 different groups. So, the top one where you actually give a mixture of microbiomes, microbes or bacteria, just to replenish the ones you lost as a result of the disease. An example here is the C. diff infection that results in the wiping of the microflora in the gut. Now you can reconstitute that, artificially. There are 2 companies out there that they're actually in phase 2, phase 3 in a development where they're using the microbiome that's actually isolated from the human fecal matter. Believe it or not, that will be approved very soon, and they will be available. Both of them will be available. There's also a situation for companies like us. How do you make money with something isolated from natural sources? That's a different story. So I think that then you have these individual bacteria. If you have a lactose intolerance, maybe you have a bacteria that can degrade the lactose, right? So there's one example there as well. So you can also treat with individual bacteria. To address the specific disease. Now, I can see very well here. And then you have these, where we stimulate the bacteria itself and generate some drug-like, properties from this. For example, the top one is an example, where you stimulate the bacteria with the oligosaccharide that seems to, generate certain metabolites that helps the disease of interest. So there are many examples here. I think this is a very exciting area, but it is nowhere, well understood class of diseases, which were often neglected because there is not enough patient pool to get money out of that. However, that's changing now because the laws have been passed a few years ago that if you go after an orphan disease in U.S. is defined as it is an occurrence is less than 200,000 patients in the entire country. And if you do go after and develop this drug for it, you get 2 advantages. One is that you get to charge a premium. Number 2 is that you get exclusivity for longer than a typical drug. And so that's an exciting area. I think we neglected this because the patient numbers are small, but I think this is going to be an area for intense research down the road, or continue to be. So here I just listed some of the indications that are classified as orphan indications and the companies they're working on. There are, by the way, some drugs already approved. I think there's alpha antitrypsin drug already approved in Europe. And there's a company, I forget, that Boston company which Sanofi bought, that is exactly founded on going after the disease. They have few drugs already on the market. So it's an exciting area, but It often involves treating with more complex way. It could be gene therapy, even it could be a recombinant DNA, or it could be directly injecting a protein. It's a complex way. But as I said, you get a premium pricing and exclusivity, why not? The ultimate goal is to either activate or inhibit this protein, right? It depends on whether you activate or inhibit the downstream effects you like to see or the effect you like to see on the disease. And so that activation of the inhibition could lead to cure. As Chuck said, in most cases, we are only treating the symptoms. Example would be, asthma. What you take inhaler, which is temporarily reduce the wheezing, right? And so HIV is an example. What you're doing, we're maintaining the disease, right? We're not treating, curing. And so, therefore, in order to make money or develop drugs, or you don't have to cure the disease. Arthritis is another example. They basically suffer for the rest of their life, but there are so many medicines for arthritis out there. And it could be a temporary relief, right? When you get a headache, you take it, and if you get it again, you take 6 hours later, right? And so the bottom line here is that even the target you chosen does not even have to cause the disease, as long as it modulates symptoms, relief. Okay? That's what we do in the case of pain. And as I said, almost all the current drugs works on proteins. Now, when you choose a protein or an mRNA you want to go after, there are certain criteria you have to have it. So, again, unfortunately, we don't have time to go through this. Make sure it is present in the tissue where you want to see the effect, whether it is in bone junctions, if you're treating in some kind of arthritis, or it's present in a heart if you're treating in a heart disease, etc. But more importantly, make sure it does not have some primary function in other tissues. If you reduce the heart rate, but if the same protein is doing something else in other tissue, right? You want to make sure that the normal function in other tissues is maintained. And I have a lot of other points here. So certainly things like, do you want to activate or inhibit the proteins to affect the effect you want? In general, inhibiting a protein is a lot easier than activating a protein, right? So remember that if there are 2 targets where you get the same effect at the end, but one need to be activated, other one is inhibited, I would like to go after the one I need to inhibit, not the activation. Activation is always tough. Often the proteins are activated using the natural molecule that are present in the body or their analogs. For example, insulin, right? You activate the insulin receptor, parathyroid hormone to activate the parathyroid hormone receptor, or even the epinephrine to activate the beta-2 adenine receptor. So that it's harder to route going that way, and but inhibiting is a lot easier. Now, last bullet highlighted here is that, okay, I've got some new targets coming out of the human genome or some other source. I think one thing you really wanted to keep in mind is that was there a success going after that class of proteins? Let's say you found a nuclear hormone receptor, that's one you want to activate. Is that something ever done? Why is it not done? Tried, failed, why? And is it something we should embark on? Phases of drug discovery, various phases. I think Chuck again touched on it, but I kind of slightly changed a little bit here, and I divided the drug discovery into 3 phases. Obviously there is one discovery phase, and then other one is development, and after discovering, there's also huge investment in selling the drug as well. It's huge, launching and selling is not easy, trivial. So 3 phases here. And again, if you start with the early on, and you have the discovery phase, research phase, and it is often done obviously in the labs like most of you working, and where you use animals and tissue culture, et cetera, and you test this and validate the targets, and even identify the leads and qualify the leads, and even do some cases optimization as well. But eventually in drug company, they all get optimized. So by the time you actually get to this phase, you would have what we call a clinical candidate, a molecule that's ready to go into the clinic, right? That is when you start the human phase 1 studies. I also listed here, if you notice, the number of years it takes typically, right? And also the number of compounds you would have evaluated. It would 5K, but sometimes, Chuck knows very well, Chuck used to do millions of compounds, screening to find one that's attractive. So, but there are also, what you call focus libraries or modeling. It comes in handy here, so you can get away with screening against a fewer molecule. Now, so I also listed at the bottom how long it takes, as I said, and then you have phase 1, phase 2, phase 3, phase 4. Phase 1 is often it is a safety study. It's a small study. It just demonstrates this molecular safety within the doses you are projecting in human eventually to treat the disease. And it is it's got dose escalation, and so start with one dose and escalate it, etc. And the safety is often it is done in healthy volunteers. Although a few cases you could do this in people with disease as well. A lot of HIV trials were early on done directly in patients. There is an argument, that look, these patients are immunocompromised. The safety requirement might be different. Therefore, we will let you do these studies directly in the HIV patients. But often it is done in the healthy volunteers, the phase 1, and often it is the students in the medical school and on students. So then phase 2, which is the early proof of concept for the biology or the effect we like to see. It's again a smaller study. Obviously, it is done on in patients, and once you also it's continuously monitor also the safety, of the individual taking the medicine as well. So the safety is continuously plays a critical role because you might see the safety signal you have seen in phase 1, because the subsequent trials after phase 1 tend to be longer, more patients involved as well. And so if you once it's proven that this drug is very effective and you go to the phase 1, which is what we sometimes called pivotal study. This is the study result for osteoporosis. The study is 3 years, 3,000 patient minimum, because where you need to show that the people taking the drug, often it's osteoporosis is an issue with the very elderly, particularly the women. One of the consequences of osteoporosis is that they break bones, particularly the hip. So FDA wants us to show that this treatment results in the reduction in the breakage of the bone, not just adding a bone or preventing the bone loss. So that's an elaborate study. On a typical study, only 2% of the patients lose or break bones in 2 years. So you can imagine to get a statistically significant results, you need to have 3,000 patients for 3 years. They're very expensive studies. We're going to talk about, we'll see what those numbers are in terms of costs, etc. And then obviously you have the FDA review that go on for a year or longer to just get an approval approval for the drug, and you could be rejected after all that, after all those. And we'll see the success rates along this various phases of drug discovery where we tend to lose the drug. And then, as I said, lastly, that you have phase 4, which is not only marketing but also expansion of the patient pool. What that means? Usually, you launch a drug for those people badly needed, usually adults. But then you have to think about people like kids, where the safety requirements are high. You establish safety in adults and move on to the kid. How about those people immunocompromised or immunosuppressive drugs? How about those people in different genetic backgrounds, such as Japanese? So there's a lot of expansion. We call it life cycle expansion. Has to be done as a part of the phase 4. That is why the drug sales not necessarily peaks in the year you launch. Usually peaks around 5 to 7 years after the launch of the drug, because you're doing all the life cycle extensions. And so the one that was concerned to the today's presentation is obviously discovery. That's where you identify target and validate. And then followed that you have high throughput screening, lead ID, which you're going to hear a lot from Chuck, and so as lead optimization, Chuck plus many others, and then you do what is called entry into human enabling studies. These are usually the safety studies as well as the scale up of the drug, because you have to show FDA not only the drug is safe, but also you have the ability to make the drug in a consistent manner. You can't just able to make drugs just for clinical trials and then you have a different quality of drug when it's ready for launch. So you're going to hear all the details from subsequent launch. I'm going to skip this, move on to target assessment. And target assessment, what the target assessment, basically it used to have two components. One is to identify some target you think it's very important for the disease, and then validate by various in vitro and in vivo studies. But then there is a third component to it that was introduced. It's called target screening. This this category was introduced since the arrival of the human genome, where you have all of a sudden 20,000 genes and 27,000 products of the genes, and which of them are really important for the disease. That is when you use genomic, proteomics, etc., to kind of identify the probable candidates. Now, as I said, the drug discovery is very lengthy, laborious, a lot of people involved. You better do a good job here, as I said. Now, what do you do in this phase? You do basically, you have a set of targets you think are involved in the disease, and you conduct in vivo studies, in vitro studies, and you develop assays because you eventually want to screen for drugs binding to this target, etc. So you do all this. You also have to develop animal models for this, right? Because you want to test the drugs in animal models before testing in humans. So this is all done as part of this. Some cases you may already have an existing model which you could use, like a cancer model, for example, you may have it. Some cases you have to come up with a new model, for example, hepatitis B. They're not good in human models. We invested a lot when I was Roche trying to fund these academic institutions and CROs to come up with the animal models. Now, again, this reiterates what I just said. You understand why this target is important for the disease. And then you also develop screens. That were basically assays. And you also develop the animal models, right? And then tractability. Tractability is a very important term. And I mentioned to you that before that there are certain types of targets we cannot develop drugs because we don't have to go in details. We're going to talk a few of those classes, but there are certain type of targets which we know how to develop. We were very successful, right? So why not go after that? So we're going to assess this target assessment. When you go to the management and say, I want to discover drugs for this target, they're going to ask, have we ever had an experience in developing drugs for this class? Why should a company invest in it? Or is there a way you can actually change that? Now, as I said, there are 3 steps now to discover a new target and and validate. You have target screening. These are the various techniques one could use for target screening, and you have a validation, identification, basically narrowing down of all the hundreds of genes you think upregulated, downregulated, as determined by genome sequence analysis, proteomics, etc. Then in this phase, you kind of narrow down. So, okay, fine. Now, all these are important, or a few of them important. And at the end of this phase, actually, you're going to select you suspect being the most important. And then you still have to validate. In other words, you have to modulate its activity in animal models and show that you can reverse the disease, right? That is the target validation. Validation is the most important steps you could take. Unfortunately, in some cases, we can't satisfactorily validate the models in preclinical studies because we don't have good models. An example would be Alzheimer's, anxiety, depression. These are the areas you have the highest rate of failure in the drugs because we don't have preclinically predicting how these drugs might behave in the human. Again, I'll come back to that. I actually have which diseases are more reliable in terms of preclinical validation. Now, in terms of validation or even target ID, you could do at different levels, right? You can modulate at the DNA level. I know where technologies I listed here. You can modulate the messenger RNA. You can modulate the protein. and even talk about, based on the high or low levels of your serum or urine markers, you could say, if you have a high blood sugar, there got to be something not right in your system. Go back and see what that might be. You have a high, insulin levels or low insulin levels, or you have a high growth hormone levels, right? So metabolomics is also very important here. It tells a lot. That's, in the past, that's how they were treated also, looking at what's, the smell of the urine or sugar levels in the urine, etc. This is very important as component of this drug discovery. So again, the techniques, remember, we won't use all these techniques on every target we're interested in. We have to pick and choose which is more appropriate based on the animal models and tissue culture models you have available, as well as the fact that the expertise you have internally. And you don't have to use all these techniques at the same time on the same target. So therefore, the various technologies you have are tools. It's up to you how you apply, how it relates to your target of interest. You pick the right one. And but what's important is to differentiate from correlation methods, which correlates to the disease. A gene upregulated in cancer doesn't mean that is causing the disease, nor it is the one that you can modulate and get the effect you wanted. That's only correlation. But there are certain methods here listed here, right? These are the methods that establish cause and effect relationship. You have to do this, one or more of these, to be able to show that indeed I can go after this target. Here is, I'm seeing an effect by modulating this target, therefore I should be able to make a difference to the disease in patient. Right? That could change depending on your diet, right? So I see a lot of diabetics around here in India. I don't think when I was growing up in India that was the case. In fact, you may have seen the New England Journal of Medicine article came out in March. The Indian woman, number 2 in the world in terms of number of obesity people. They were 25 years ago, 29th in the rank. That's amazing statistics. Remember, obesity leads to heart disease, diabetes, renal disease. It's alarming. I know I used to go to the functions in Indian functions. Every time I come, I see it. That's one line for the no sugar, the other one for with sugar. I think it seemed like more people standing for no sugar than the line with sugar, which is very alarming, really. I think it's all going to change, even though you are genetically prone to it, but you can control it. So the next, I think, hopefully, I convinced you that there are many methods one could use to validate. But what's important is the validation in preclinical animal models is very essential. And but there is certain diseases where the preclinical models are not reliable, particularly in the CNS area, and some extent in oncology as well. And so there we have to take a risk and move forward. So many CNS, central nervous system disease, targeted drugs failed in recent years. You may have heard it that the Biogen stock plunged $9 billion overnight because one of their MS drug failed. It was only 2 weeks ago, the news was. So they're betting a lot on it, and Roche had many drugs as well in pipeline, but they're still waiting to see some success in that. Now, I think it makes, hopefully appreciate the risk in the drug discovery, and hopefully that encourages you to do a good job at target validation. So the next few slides actually talks about the risk. So what I'm depicting here is the number of drugs produced over a period of time. And you could see that, I mean, we are reasonably doing good job until 2005. So we dropped to the all-time low in the number of drugs produced. Although we picked up a little bit in recent years, actually we're doing pretty well last 2 years, very well. But then this is a slump here. That created some nervousness among pharma. Okay? Now, that's the same data actually depicted slightly differently here, where it's a breakdown into the drugs produced by the typical pharmaceutical company versus those produced by the biotech. You see that biotech began to produce more drugs than the traditional pharmaceutical companies, which is very important to see there. Now, now the other way to put this risk, what that risk for pharma if you produce fewer drugs, here an article that came up last year in Nature where they looked at the revenues generated in the 7 years following the launch of the drug and compared to the money that went into producing the drugs in the preceding 7 years of launch. So obviously the one or more is, more than one is basically in the profit side, and less than one is, you're losing money on the drug. So that coinciding with the slump in the drug, you see that the ratio began to be very close to 1. And we actually, subsequent years, we have been losing the money, although the drugs were launched. And although, as I said, with the advent of many new drugs, actually our ratio began to go up. But you remember, pharmaceutical companies really enjoyed very good ratios, very good profits, not anymore, and because of the high attrition rates. Now, also interesting to see that the ratio is variable depending on the indication what a company is interested in. So you are still on the positive side during this period if you're working, if you're working on indications like endocrine indications, but on the other hand, cardiovascular and central nervous system, etc. This is for the last several years. I think it's 10 years or so. And so we began to lose this money in some areas more than others. Now, it's interesting to see that how many drugs a typical company, big pharma, produces in a year, and on average we only produce 1, sometimes 1 to 1.5. You see that there's some companies were more successful than others, but an average is 1 to 2. In order to produce 1 drug a year, a typical pharma maintains as many as 150 to 200 projects running at various phases. Could be preclinical, could be phase 1, phase 2, phase 3, etc. So, in other words, we actually engage in developing so many molecules, but we only get 1 drug a year. So we're losing money on all the failure drugs. If you look at why they're failing, I think one of the reasons is that poor target validation. Really, that's one of the reasons. It's not the only reason, because that poor target validation shows up in phase 2 when you're trying to generate some safety data. Also could fail in phase 1 where we're trying to show some safety, because this target actually has other functions. We call it target-related toxicity. By AstraZeneca putting their data as well as some other companies' data where they're showing that what's the success rate going from phase to one phase to the other. So obviously the worst in terms of survival of the drug is going from phase 2 to phase 3. Phase 2, what is that phase 2? Which is showing the efficacy, early efficacy in people. If we had done good validation, perhaps you wouldn't have been waiting to see this, in phase 2. So overall success, if you have a drug from beginning to here, it's from entering into the phase 1 to the launch, is 7%. Even if you put a molecule in phase 1, you have chances of that becoming a drug is 7%. So we lose a lot of molecules. Now, this is the most important, or rather most expensive part of the drug discovery. It is not the preclinical. Preclinical is cheap because you deal with cheap scientists and animal models and in vitro models, etc. It is the expense goes up exponentially when you start using human subject. Help to deal with tough targets, etc. But I don't think it changed the overall timeline. Most of this increased cost and increased timelines comes from the fact that you have to do these trials longer, both from the perspective of the safety as well as efficacy. I don't have time to go through this, but one thing I can say is that all this high attrition, high cost, and is as a result of tackling indications that are traditionally considered high risk, like oncology, or CNS, where we don't necessarily understand the biology very well. We don't, as I said, there are no good animal models either. So high risk target these are, therefore we're taking the risk and end up getting fewer drugs, investing more money. As a result, what happened was about 10 years ago, all these pharmaceutical companies began to do a few things. One was they laid off all the internal R&D, because they adopted a system where why not rely on the external innovation by biotech companies, even some academicians. And the other thing they decided to do was that, internally keep the people who are necessary to do the core jobs. When you bring the molecule in-house, you should be able to develop it, etc. And so New Jersey, that's considered the world capital of the pharmaceutical industry historically, lost more than 200,000 pharma R&D scientists in the last 7 years. This also includes as a result of the mergers and acquisitions. So we lost a lot of. However, pharmaceutical companies still kept the R&D budget intact. Instead of spending internally, they're spending externally. I'm going to talk about this on Sunday, how they're spending the money, right? Which the good news is that it's still their R&D money, but it's not for internal R&D, but rather externalization. As Chuck mentioned this also, if you look at the per employee, the amount of money spent on R&D, pharma is on the top. I think it's 130,000 per employee, among any industry. Yeah, here's the point I wanted to make. A lot of drugs gets withdrawn for various reasons upon launching. So it's also important to recognize what reason they get withdrawn, because it helps to understand or de-risk early on. If it's a cardiovascular, a lot of drugs are withdrawn because of the QT prolongation, right? And if you had done thorough job preclinically, maybe you could have avoided even launching, developing and launching as well. That is why the information that is coming out of these withdrawn drugs is as important for a drug industry. Very important. They lose the revenues, obviously, like Vioxx, they lost $3 billion a year, right? But there's a lot of learning to de-risk the pipeline, right? So, but it's given that I had a slide that where all the drugs withdrawn the last 10, 20 years, I'm not sharing with you, but you could find it online as well. Amazing that you get withdrawn 1 or 2 drugs, blockbusters a year. This is a slide that talks about the reasons for withdrawn, right, as a function of various properties of the drug. And here is the clinical efficacy and efficacy in phase 3 and formulation. Drugs could fail many reasons. But what's important for this discussion today is that is there something could have done to prevent this? Is there something could have done to validating the target or finding the right molecule? Yes, as I said before, that poor target validation manifests itself in phase 1, phase 2 in the form of safety and efficacy, right? So you already brought it all the way to phase 2 to realize the drug is not quite as safe as you thought or as efficacious as you thought. So it's very important that you could choose the right target and validate it carefully. You can avoid this attrition rates. Now, so this is going to stop after this. Basically, hopefully I emphasized enough that target validation is very important, very important. Not every drug makes money. The only 10% of the drug makes 90% of the profits, right? And so I think we can change this if we do a thorough job of target validation, and it's increasingly hard to get blockbuster drugs. The failure rates are getting worse and worse. Having said that, all this high attrition really manifests itself in the drug pricing. You see a lot of talks about drugs cost too much, etc. That is true. They cost too much than ever before. But look at the fraction of the healthcare cost that goes into buying drugs for a patient. It's only 10%, believe it or not. It is the doctor, it is the infrastructure, it is the insurance companies. That's where all the money goes. It's only 10% drug cost. Now, drug companies historically enjoyed very good margins up to 40%. Now it's down to 20, 25%. It's still pretty good, I think. But remember, you have to allow those kind of profits if anyone wants to invest in this business, take a risk, and take a risk and accepting the risk. So I think you could supply drugs very cheap, but longer time innovation will going to affect. Particularly the indications like Alzheimer's, anxiety, depression, schizophrenia. You're not going to have drugs if you don't invest in it.